Thank you, Heidi, for that introduction. Can everyone just give me a raise your hand, or a few of you raising hand, you can see everything well? Yep, excellent. Okay, I'm just going to put your hands down. All right, we're going to get started. As long as here we go. So firstly, I want to get an idea of who has used a diagnostic service, who has sent plants to a diagnostic lab for the purposes of identifying a pest or disease problem. So I'm just going to launch this poll. Hopefully that's, yep, so we're just collecting the responses. So click the box that best applies. Yes, I've sent a sample many times. Yes, only once or twice. No, but I've thought about it. Or no, I always know what's wrong with my plants, or just no. A few more seconds. 5, 4, 3, 2, 1. Okay, I'll close the poll and share those results. So about half, many times, that's great. Another quarter, oh, a bit, that's great. All right, that gives me a good idea of people. All right, hide these results. And moving right along. So it's a short talk, just split it up into two. First thing, through the talk, there are going to be some images on the right-hand side, mainly. At the end of each slide, the disease or the pathogen that's causing those diseases are going to pop up. So if you want to, you can guess what pathogen is this that's causing those symptoms, and at the end, I'll ask you who got them all right, and hopefully it'll be fun. Anyway, so firstly, that's the first lot. Oops. So what's a pest? What is pest and disease diagnostics? Simply put, it's identifying pests and diseases. So we receive sick plants, we run tests, and we get some results to assist in working out what's wrong with them or what's not wrong with them. Because sometimes knowing what's not wrong, even if you can't work out what is wrong, knowing what's not wrong is valuable and can help you to work out what to do next. We interpret those results on our report and we provide recommendations wherever possible. So while not diseases, this is Sarah, Sharon and Joy, and these are just some of the people who work in Grow Help. Okay, so why is it important? Hopefully most of you know. However, many diseases have similar symptoms to each other, and we have to run specific tests to work out which disease you have. And knowing which one you have can help you understand the biology of the pest, know the high risk periods, and proactively manage those diseases, as the management for one disease may well be different from another. And the idea being to break the life cycle or make the growing conditions less favorable so you can put in the most appropriate management actions. Even for pesticide application, or in the cases of some pathogens like Fusarium, the value of fungicides is pretty limited. So it can help you to make decisions for future plantings, for this planting, and so on and so forth. Just trying to move. Okay, so what do we receive? What do we detect? We can test for a really large range of pests, for fungal pathogens, bacterial pathogens, viruses, nematodes, and some groups of insects and mites, just depending on where we have taxonomic expertise. Where we don't, we've been improving our ability to use molecular tests to identify insects. And this works sometimes, but doesn't others, unfortunately. So we're using some traditional testing techniques where we take the margin between the relatively healthy and the unhealthy tissue. We're putting it onto selective fungal media or bacterial media. We wait to see what grows out and then identify the organism that's grown. hopefully consistently. And in this case, we got a particular pathogen, which you'll find out in a moment, very consistently. And it's awesome to get those cut and dry results, but it's not always the case. And sometimes we get results that are very tricky. We also run tests for specific groups of species, or like a genus level, or for particular species. And that may be for virus groups or bacteria or some fungi. Sequencing is where we send off a particular gene region to a third-party provider, and they provide the order of the DNA in that gene region, and we compare that to the sequences, the library or the database that's online where the organisms have been identified, and we can compare our DNA to the DNA that's online and infer an identification. So there are quite a few things that we can do to detect pathogens, and these are just a small subset of the types of hosts we receive regularly in Grow Health and the types of pathogens that we are most often identifying. This is by no means a complete list, and particularly in production, nursery crops, we will get a really wide range of veg as well as other crops, and oftentimes we have never even heard of a plant, and we go, oh, okay, and we have to work out what's wrong with it. It can be challenging. We also end up providing some indication where we suspect that there are non-pathogenic disorders, whether it's edema or a suspect herbicide or nutrient deficiency or disorder. We don't have tests to determine whether there's a nutrient deficiency or a phytotoxicity, whether there's herbicide damage, but there are other labs that do that sort of work. We can look at it and go, we think it's that, or we think it's so on and so forth. Does that match up with your observations, so on? Okay, next bit, how long does it take? How much does it cost? How do we submit? And the pathogen on the right is bacterial speck. All right, so how long does it take? And to a certain extent, it's dependent on the sample that we receive. Some samples are really straightforward and they're awesome. We love getting them. And you love having us be able to identify it easily. We get something like broad mites, we can look at it straight away, go, it's this, and it's done. And we can get those results back to you, hopefully on the same day. Sometimes it is the next day. Other things, powdery mildew or downy mildew, we can go, okay, we've seen these things, or we haven't seen these things, if that's what you're concerned about, and then take the sample to the next step. Where we have to culture out a pathogen, it can sometimes take days is normally the minimum. A week is usual to get that preliminary ID. Sometimes these things can take many weeks, and we don't often wait many weeks to culture and mature that fungus or bacteria. We'll more likely to go, well, we think it's probably this. What do you want to do? We can sequence it. It's going to cost this amount. Or the other result, we can wait for other results that we might be also waiting on, depending on the sample. So it is a complex. There is no one size fits all. It will take 2 weeks. It really is dependent on the host, on the symptoms, and sequencing. If the external provider with COVID, it's been taking longer. And we have sometimes taken longer with COVID as there have been staff away. Okay, and the pathogen on the right is CACV. We will often get samples, soil samples for testing for Phytophthora particularly. We can also detect Pythium and Phytopythium with the same method. This type of testing takes a couple of weeks. So we only set it up once a week. It takes a week to have or about 5 days to have the symptoms potentially develop on our baits, which are blue lupins. And then we put these onto a selective media. It all takes time. Synthesizing the data also sometimes takes time, particularly where we have many pathogens detected or many potential pathogens detected. And potentially some of them are secondary, some of them may be interacting. You may have multiple primary pathogens. And then we have to research, well, what is the host? What is the pathogen? Are they producing those symptoms that are normal for that pathogen? And it isn't always straightforward, unfortunately. The cost is normally, on average, it's about $300 a sample. It does depend on how much work is involved. If we have to run a lot of tests to come to a conclusion, then it will cost more, but that is on average. If you are wanting to send in samples for, let's say, indexing to get market access, then just contact us and we can provide a quote or an indication of how much it's going to cost more specifically. And with that said, all production nurseries receive 6 free samples per year, as we have basically it's contract rates, a project that's funded through the nursery levy and Horn Innovation and us. So if you have, or let's just raise your hand, who has a nursery on site that you use, you grow seedlings, plant those plants? Anybody? I'm not seeing any raised hands. Well, I know some growers do have their own nursery, so if you can send us samples, then you'll get 6 free per year. Just moving to the pathogen causing this one, Pythium, parsley. Okay, so we have, in terms of how do we submit a sample, we have a lot of information on the website. Choosing the right sample is important. So if you have any doubts, you can send us an email, give us a call, and we'll help you. Really what we want are samples that are in the process of actively dying. That might sound interesting, but actively dying. So we don't want dead plants. Dead plants have all sorts of secondary bacteria, fungi. We don't want those. We want the plants that are in the process of dying, where we have the margin between dead and dying tissue. For plants that are smaller, we need more plants. Packaging the sample is important. We need to keep it dry, so include paper. If you're putting in Ziploc bags, you can put in paper around it. You want to make sure that you contain the growing media or the soil so that it doesn't go throughout the sample. Like this last sample here up on the right, it's not fun. It's much nicer when we receive a sample like this, and you're more likely to get good results. So ice blocks produce condensation. Water tends to break things down in the sample. Make sure you label your sample. It sounds really obvious when you say it out loud, but we get samples with no identification, and we just have to sit them in a room and hope that in the next couple of days, someone owns up. Sometimes we go, no one, oh, what about that sample? You got it a couple of weeks ago. Probably not going to get the best results. Watermelons, gummy stem blight. Okay, so we have an online submission form and you need to complete that for your samples. Hopefully that will be changing soon, the form, we're trying to upgrade it, but it's been a process. It's best to send samples by courier on a Monday or a Tuesday. Wednesday can be a bit dicey, particularly if you're using Australia Post. COVID has really impacted transit times, so it is safest to send on a Monday and Tuesday. I think I'm just about running out of time, so there are a lot of things on the website. So if you search for Grow Help Australia, you'll get a lot of information. The landing page is here. And thank you, everyone. So lastly, who managed to get, did anyone manage to get all of them right? Raise your hand if you managed to guess all of them right, or even maybe half. How about half? No halves? Okay. And I'm just looking back on the poll. No, I know what is wrong with my plants. I always know what's wrong with the plants. No one hit it. So at least you're being honest. That's great. If you have questions, you can always send us an email at that address. Over to you, Heidi.
Fantastic.